Dear friends and colleagues, I wish you all very welcome to this 90-minute webinar on state-of-the-art in anaerobic digestion modeling. This webinar is a joint effort of two of the specialist groups within the International Water Association. The one on modeling and integrated assessment, which we call MIA, and the specialist group on instrumentation, control, and automation, ICA. My name is Dr. Ulf Jeppsson from Lund University in Sweden. I'm the chair of the specialist group on modeling and integrated assessment within IWA. And before we start the actual webinar, I want to take this opportunity to say just a few words related to the work of the MIA specialist group. A short introduction of the ICA specialist group will also follow. The MIA specialist group is, or we try at least, to be one of the most active specialist groups within IWA. And we are the group that really focusing on modeling of all aspects of water and wastewater processes and everything related to that type of modeling. It's open for everybody who's a member of IWA. And we currently have some 1,900 members in the specialist group. One of the most important ways that we work through is by task groups. A task group is a small size group of experts focusing on a specific topic for a number of years and then normally producing an IWA scientific and technical report. Currently, we have 5 active task groups under the MIA umbrella. You see them here to the left. Out of these 5 active groups, we actually have 3 that are almost finished and will soon deliver their final reports to IWA. Secondly, we are also active through working groups. A working group is normally a larger group of persons promoting and investigating a more general topic. And thirdly, we reach out to via our 2 main recurrent international conferences, the WRR-MOD and Watermatics conferences. And speaking of conferences, our next main event is the WRR-MOD 2023, or 2022 plus, as it's sometimes called, the modeling seminar in Stellenbosch in South Africa, which will take place the 15th to 18th of January 2023, and is organized by Dr. David Ikumi and his colleagues. Then in the late summer of 2023, we have the Watermatics 2023 Modeling Symposium, which will take place in Quebec City in Canada, and being organized by Professor Peter van Rollegem and Dr. Elena Torfs. If no one hear about what we are doing and what we achieve, then we hardly exist as a specialist group. Therefore, we have a number of channels for you and everybody else who wants to learn more about our activities, to follow news and updates, and so on. We have the official MIA website at IWA Connect. That is available for all members of IWA. If you're not a member of IWA, you can also find the similar type of information at our open website, which is available for anybody to go into. We also have our LinkedIn and Twitter, and we also produce a regular newsletter for our members. And finally, we have our own YouTube channel where we make webinars like this one generally available. So I hope to see you all as members, if you're not all that already, in the MIA specialist group in the future. With that little intro of the MIA group, I leave the floor to Dr. Angel Robles from University of Valencia in Spain. Angel Robles is also a member of the management committee of the ICA specialist group, and he will give a short introduction to that specialist group and also present the topic of today, the AD modeling webinar. So I wish you all a fruitful webinar. Thank you.
Can you hear me now and see me, Wolf?
We hear you now, Angel. Please go ahead.
Okay, thank you. Well, thank you. Well, I will want also to welcome everybody to this joint webinar within the MIA and the ICA specialist group. And of course, I will want to make some quick introduction on what we do from ICA, and just in case you are not very familiar with this specialist group, to let you know where you can find also more information about it. Well, my name, as Ulf introduced, is Angel Robles. I'm the newsletter editor and public relations coordinator in the ICA Management Committee of the ICA Specialist Group, which is currently, today, is chaired by Juan Baeza and co-chaired by Chris Villeneuve. And I emphasize today because we are currently in the process of renewing the Management Committee. And unfortunately, this has been the last cycle for Juan and Chris. So I will want to start also by deeply appreciating the great contribution and also the great contribution from the rest of members from the Management Committee that are leaving the group very soon. The ICA Specialist Group is an international forum to exchange knowledge, methodologies, and experience on all aspects of instrumentation, control, and automation within the whole water cycle. So any IWA member is very welcome to join by visiting our space on IWA Connect by clicking this button, as you know. And now we are around 1,400 members and 14 management committee members, including 5 affiliated young water professionals in the management committee. The main objectives, our specialist groups aims to promote the use of ICA, and we try to show how it can help to create or develop more sustainable systems, as well as systems with lower management problems. In fact, ICA should be used to increase safety, reliability, and performance of any process in general, and of course within the whole water cycle. We are also very concerned about training and education of ICA, since specific training will help water professionals to get better capabilities in solving the typical problems that appears in these processes. And the key activities of ICA can be summarized in this slide, some of them. One of the key activities is the publishing of our newsletter that can be found in the specialist group IWA Connect page if you are interested in. And we publish this with a rate of 2 issues per year. We're also very involved in organizing and supporting any conference related to ICA and workshops, and of course, as is the case, webinars, supporting also related task groups and working groups and clusters, and among others. So in this case, we encourage you also to join our next ICA conference that will be held on Beijing. This is not completely confined if this conference will be in virtual format or not. And we encourage you to be connected in IWA Connect, to be aware of any new related to this, and also to join 2 upcoming webinars on advanced biological nutrient removal that will be announced very soon. So please don't forget to follow us in the IWA Connect to be updated. So let's start with today's webinar by introducing very quickly anaerobic digestion technology. And as most of you know, anaerobic digestion AD is a multiple step process in which complex organics are converted to the most oxidized forms of one atomic carbon, that is carbon dioxide and methane, and the initial processes are conducted by bacteria that are the responsible of hydrolysis and fermentation, and the following ones are mainly carried out by syntrophic associations between bacteria and archaea for the anaerobic oxidation of alcohols and organics to these more reduced monatomic carbon forms. Nevertheless, besides these biochemical reactions, other mechanism schemes might be taken into account for enhanced modeling practices, that is the case of today, taking into account, for example, the sulfur cycle that interacts very strongly with the phosphorus cycle, the methyl oxidation reduction cycle, or even the action between the electron donors such as methane and hydrogen. So enhanced modeling practices will help to deeply assess the potential of AD as key technology within a circular economy. Indeed, AD helps to mitigate environmental impacts associated to the depletion of different resources, such as nutrients, fossil fuels, and even related to water scarcity. Indeed, AD is a key technology for the sustainable management of a wide range of wastes, including food waste, manure, or even urban wastewater. For instance, when compared to aerobic processes, AD avoids the energy demand for oxidation organics and produces methane as energy source. Indeed, the anaerobic digestion has been classically classified as a default process for bioconversion of organics to renewable energy and biofuel in the form of methane, but it should be also looked at a driver for other purposes, such as nutrient conservation and recovery, a driver for value-added chemicals production through mixed culture biotechnology, and all this keeping low energy demands, low greenhouse gases emissions, and low sludge productions. To widen this applicability, new processes are being also explored and must be also modeled and optimized. And here we can see a picture, and I'm not going to be in detail because Demi and Aston will be show it later. So, well, just in case, just you can see the wide range of applicability regarding the hydraulic retention time and the solids concentration of several technologies available in the market. However, of course, there is always a however. Some giants might be still evaluated and modeled, of course, for generating accurate tools able to predict the performance of AD in different conditions. So given the opportunity to practitioners and also to research to optimize and to evaluate the optimal conditions for AD implementation. Among others, we could highlight the increased importance of phosphorus, sulfur, and methyls as electron source. Also, the need to consider variable mass volume contents during high solids anaerobic digestion, or to consider trace elements, complexation, and precipitation. And why not to account for the membrane interactions in high-rate membrane-based systems. Last but not least, of course, to enhance the monitoring and control systems for AD optimization. Finally, I will want to take the opportunity to encourage you also to join the next AD conference that will be held in Michigan next year. In this case, if the situation allows, it will be physical, it will be a real conference, not virtual. So please follow the IWA Connect and the IWA network to be aware of upcoming announcements on this conference. And that's all. Yeah. Well, let's move to the agenda. First of all, some remarks. Remind that this session is being recorded. Of course, the microphones and cameras has been disabled due to the large number of attendees, and the normal chat has been disabled. Please, any question that you think that could be interesting for the discussion at the end of this webinar, please use the Q&A tool, and we will do our best to answer them during the session, writing or orally at the end of the speakers. And we will start with the first speaker, that is Raúl Muñoz. And Raúl Muñoz is a professor from University of Valladolid. He is chemical engineer also by the University of Valladolid and doctor in environmental biotechnology by Lund University in Sweden. And currently he is known and he's renowned expert on biological processes for the production of biogas, gas treatment, biogas upgrading, or microalgae application for wastewater treatment. So please, Raul, the floor is yours.
Thank you very much, Angel, for this kind introduction. Okay. Okay. Can you see the screen?
We see it, Raul. Go
Okay,
ahead.
perfect. Okay. So first of all, I would like to thank the organizing committee of this webinar for this kind invitation to provide a summary of the workshop that we recently organized in Valladolid. It was entitled Anaerobic Digestion Quo Vadis. So good morning and good afternoon, everyone. So on October 21st, the Anaerobic Digestion Quo Vadis workshop was organized by the Institute of Sustainable Processes of the University of Valladolid and Cretus Institute of the University of Santiago de Compostela. Just a bit of context about the workshop. There was 30 years ago, there was another workshop organized in Valladolid and sponsored by the IWA on anaerobic treatment technologies for municipal industrial wastewater, and we wanted to provide an updated version of this workshop. Okay, it was held virtually and in person. And basically, we were aware that in the last 30 years, the scope of anaerobic digestion processes have rapidly expanded from a simple wastewater treatment technology to a platform capable of producing renewable electricity from organic waste or energy crops. And in the latest years, anaerobic digestion is regarded as a useful platform for a multi-product biorefinery. In the last years, also new tools and processes such as molecular biology, process automation and control, biogas upgrading, nutrient recovery, or even organic acid generation have upgraded the potential of this technology and also increased its robustness. The speakers of the workshop were Jules Van Lier, Cornel Ravet, Anna Suarez, Irene Angelidaki, Jean-Philippe Esteller, and Lutgardi Raskin. Okay, Jules Van Lier presented an overview on the potential of anaerobic membrane bioreactor for the treatment of complex chemical wastewater. He presented a case study treating a very refractory and toxic wastewater, mainly composed of aromatic hydrocarbons at a high salinity, around 60 grams of sodium per liter, high temperature, almost no nutrients to support microbial growth, and no trace elements. Okay. They implemented membrane anaerobic bioreactors in order to prevent the washout of degranulated and deflocculated biomass due to the toxicity of the wastewater. Anaerobic membrane bioreactors support long solid retention time that enhance specific catabolic conversion of bacteria to degrade these toxic contaminants. They allow an easy in situ bioaugmentation of a specialized bacteria in the reactor, and also the solid-free nature of the effluent allows for an easy process reclamation. And this is basically one of the main graphs showing the potential of anaerobic membrane bioreactor compared to conventional USAV reactor for the treatment of a wastewater with 0.5 to 1 gram per liter of phenol at a high salinity, where anaerobic membrane bioreactor supported an almost complete removal of phenol compared to USV reactor that show a non-complete but rather improvement on phenol degradation. Cornel Ravet also discussed a case study on caproic acid production from thin stillage in a presentation entitled From Anaerobic Digestion, from Limitation to Potential. So he presented initially a technology based on suspended growth bioreactors, okay, 2 different configurations with ultrafiltration, supporting a downstream based on microbial electrochemical cells to produce caproic acid. The main results of these experiments show a good product stability and a good composition of the caproic acid. But unfortunately, the production rates and the price of the caproic acid was not competitive, which require further improvement of this technology. They explore the potential of granules, okay, which allow, uncoupling liquid and solid retention times, support higher biomass concentration and therefore higher production rate. And by using granule-based bioreactor, they managed to increase the the caproxylate production rate in grams per liter per day up over 25 grams per liter per day of butyrate and over 15 grams per liter per day of caprate. So based on this research, they managed to have rates of butyrate and caproic acid production over 1 gram per liter per hour with a pretty good selectivity using pure cultures. They managed to have a very good downstream of this caproic acid, but they didn't manage so far to have all this performance all together, and the market for this product is still not strong enough. Ana Suarez provided an overview of the state of the art of nutrients management and recovery from digestate. Okay, she focused on advanced anaerobic digestion concepts, which increase solid hydrolysis, and therefore biogas production is increased, but also the nutrient concentration in the diastates, in the water and liquids, is also increased. As an example, the water and liquors from a thermal hydrolysis process coupled to anaerobic digestion exhibit ammonia concentration in the range of 1,200 or 1,000 to 2,500 milligrams per liter compared to 500, 1,000 milligrams per liter of conventional AD, the water and liquors, and also total phosphate concentration up to 200 milligrams per liter compared to the total phosphate concentration in conventional liquors from 20 to 100. She discussed the main results on some pilot testing at Cranfield University, treating 10 cubic meter per day of water, where ammonium was recovered at 10 minutes of contact time using zeolites with good efficiency, and also phosphate was recovered using a hybrid anion exchange at a high recovery. Unfortunately, the current panorama is not very promising. Okay, she discussed the 2 plants of estruvite in the UK, none of them working. There are also only few estruvite plants in Europe, which are the exception. And unfortunately, the potential of estruvite recovery, even if the technology is ready at high TRL, it can be only applied to less than 10% of the wastewater treatment plant. And analyze the need to find the best option to recover the product at a local level, and to have a very clear business case and LCA at large scale in order to improve this panorama. Irini Angelidakis focused on moving beyond biogas. Okay, she considered anaerobic digestion as a treasure chest that is not only capable of producing heated electricity from biogas, single cell protein, biomethane, methanol, aviation fuels, biodiesel, chemicals, algae, and even biodegradable plastics can be produced from biogas. Okay, she focused in one of the technologies. She's working more intensively at DTU, which is hydrogenotrophic biogas upgrading, based on reducing the carbon dioxide from biogas using hydrogen produced from water electrolysis, using the surplus of renewable energy. Okay, in 3 different bioreactor configuration: ex-situ configuration, where hydrogen and biogas are upgraded in an external bioreactor; in situ biogas upgrading, where hydrogen is injected into the anaerobic digestion; and also an hybrid process, where biogas is partially upgraded in the anaerobic digestion and then finally polished in an external bioreactor, also provided with hydrogen. Jean-Philippe Estegier also provided an state-of-the-art of instrumentation, modeling, and control in a presentation entitled "An Old Story for Today and Tomorrow." Okay, Jean-Philippe encouraged the audience to use a more smart sensor using this development at his laboratory on near-infrared spectroscopy, which shows a very good prediction between the measurement of this technique and fast biochemical methane potential test. He also described the evolution of anaerobic digestion modeling, okay, that has experience and exponential growth over the past 15 years, mainly due to also to the publication, after the publication of the ADM1 model. Based on Jean-Philippe experience in control of anaerobic digestion, he also suggested physiologics and adaptive control as the best control strategies, exhibiting a good performance, a reasonable mathematical complexity, and also a reasonable instrument complexity. Finally, Lutgardi Raskin also reflected on engineering on microbial communities, okay, to improve the performance of anaerobic digestion and also on biochemical production. And she basically approach two different strategies: top-down design and bottom-up design. When using this top-bottom approach for engineering microbes, one should use the most complex microbiome as inoculum to have high diversity, select the proper bioreactor operating condition to obtain the desired outcomes in the process, apply modeling, adjust the process variables in continuous bioreactor to influence or to tailor the function and to obtain an engineered microbiome, iterate to optimize the process, and finally do a techno-economic feasibility evaluation and also an environmental impact evaluation. It is also possible to use a bottom-up approach to engineer microbiomes. Okay, first isolating and sequencing genomes of the desired microorganisms, use metabolic modeling to obtain and to design the desired outcomes, use gene editing and synthetic biology to optimize the pathways, and finally combine the microbes to obtain an engineered microbiome. Iterate and finally do a techno-economic and environmental evaluation. Okay, and this is basically a brief summary of the workshop held in Valladolid 1 month ago. So thank you very much.
Thank you very much, Raul. Thank you for so nice summary of such an interesting workshop that I personally joined. I enjoyed a lot. So congratulations also for this workshop, and of course for this presentation. So our next speaker, I'm going to show you there. Well, just to remind you that please you can use this Q&A section for making any question for the panelists. So please don't be shy, use it. Now we are 166 person of us, so we are a high amount of people that of course has a lot of questions and interesting questions for the discussion. And well, let's move to the second speaker. Is Deputy Director of the Australian Centre of Water and Environmental Biotechnology at the University of Queensland and Professor in the School of Chemical Engineering. Damien Baston has over 20 years experience in wastewater process modeling, design, and environmental biotechnology. And he's going to give us a talk about the ADM1 model. So please, Damien, the floor is yours.
Thank you, Angel. Okay, so as Angel has said, I have approximately 20 years digester modeling. It's actually 24 years because for 3 years I worked on a different model. And then for the next 20 years, I worked on the ADM1, I guess. So we published this in 2002. It was actually an IWA sponsored task group, one of the very early ones, certainly one of the first ones after the ASM was produced. So one of the most common, the ASM series, the activated sludge model series, was the most successful task group at the time, of course, because it's one of the only task groups, but it has also since been one of the most successful. And one of the most common questions they got was, this activated sludge model is very nice, but what about the anaerobic digester? And actually, none of the people who did the activated sludge modeling were anaerobic experts. So IWA put together a group of experts as well as one recent PhD graduate, it was myself, to work on this. And we worked on it for about a bit over a year. And we had a conference, we had a workshop in Switzerland, which was organized by Hans-Rudi Siegrist, and we put together this model. It was then very, very successful. So it was presented in 2002 at AD9 in Belgium, and it's got very little that's new, and most of what was new was not very good. So actually, I'll talk about the model structure soon, but the best stuff was the stuff we took from all the models that have been done previously. It was cited more than 2,000 times, and it actually sold out. I think the first run of 500 copies sold out about 10 years ago. So ever since then, you'll be getting print-on-demand copies of the ADM1. And it's probably cited in about 60% of modeling papers. So I'll just talk about what we put together. So most of the bottom end of the model is common to all models. My pointer is not working, but that's okay. So from this point, you can see my cursor, I think. From the carbohydrate, protein, lipid part down, this is very common to other models. We decided to include propionate, butyrate, and valerate because they show different responses. It wasn't supposed to be a model that's good for sludge digesters or a model that's good for high rate digesters. It was focused on, I guess, activated and primary sludge digestion, but it was intended to be useful for basically all engineered anaerobic environments. Now, I'll just see if I can get this working. Yep, okay. So as part of the task group, we decided to make hydrolysis a two-step process where particulate organic material, which we called complex organic material, XC, would go through a first hydrolysis step to form inert particulates, inert solubles, lipids, proteins, and carbohydrates. And then these would then hydrolyze in the second step to sugars, amino acids, and long-chain fatty acids. Now, that was reasonable because this particulate material was a composite material. And it would undergo a depolymerization, decomposition process, but it turned out to be a bad move because essentially it doesn't allow you to independently characterize this complex material, and the feed materials actually consists of a lot of different materials. So it made this into a two-step process, but is with just a one-step process. And generally now we characterize the input material in terms of these components, which we then apply a common hydrolysis kinetic to. Sorry about all the red ink. Okay, so just talking about how things move. So when we published the model in 2001, these were the kind of digesters that were out there. So we had high rate anaerobic systems, we had liquid mixed systems, and we had anaerobic lagoons. All of those have been modeled using very similar models. In the period since then, we've seen a lot of processes emerge. So we've seen anaerobic MBRs, we've seen high rate thermophilic systems, plug flow systems, right up to solid phase leach bed. The ADM1 is definitely applied to UASBs, EGSBs, etc. So it's applied in many different ways, but the simplest way is just to extend the solids retention time. A better way is to treat it as a fluidized bed reactor with the ADM1 handling the organic conversion. There's been quite a lot of papers on an application of the ADM1 to UASB and EGSB reactors, and it's fairly successful. It handles the acid overload, the failure mode in UASB reactors fairly well. And just speaking about failure modes, so each type of these reactors has got different failure modes. So a liquid mixed digester, its failure mode is either loading it too much flow or essentially ammonia overload. Whereas a high rate anaerobic digester, the failure mode is acid overload, where you exceed the capacity of the methanogens to remove acid. So the ADM1, unlike focus models that are in, for example, BioWin or Sumo, which are focused on the sludge line, the ADM1 was intended to be able to handle different reactor types. And it handles them reasonably well. So you have to make some modifications, particularly mainly not to the biochemistry, but mainly to the hydraulic implementation to apply the ADM1 in a high rate anaerobic digester versus a liquid digester. But essentially the same model can be used across this whole domain and has been applied across this whole domain. So ADM1 has been applied in all of these types of reactors. Okay, so I just wanted to talk on what people find the hardest, which is input characterization. And there's a fair bit of experience in this. So you can get the ADM1 in MATLAB format from Lund University or DTU. You can get it in Aquasim format from me. You can get it in, you can, I think there's a Sumo implementation. There's not a BioIn because it's not state compatible, but there's a GPSX implementation. So you can get versions of the ADM1 that are either free or within the package that you're working with. You can get Python versions, Java versions, etc. The difficult part is then applying, and the most difficult part of applying it is the input characterization. So a lot of this is based on experience and knowledge. As I said, we don't use the XC parameter anymore, or the XC as an input. We basically classify it as carbohydrates, proteins, and lipids. We characterize the protein component based on the nitrogen content in there, and then we split the lipid carbohydrate based on the COD mass ratio. So lipids will have a very high COD, whereas carbohydrates have a low COD. And then you have to estimate essentially so that gives you the gradability and the different fractions. You have to then estimate the weight. And we can do that in two ways. We can either do an anaerobic batch test, and if this moves up rapidly, we can fit the parameters, the degradability, the carbohydrate protein lipid ratio based on the methane response during batch. It's normally a bit conservative, but realistic. Or we can fit it based on continuous data using parameter estimation. So looking at the parameters you obtain from a batch test versus the parameters you obtain from a continuous reactor versus the parameters you might obtain from a full-scale reactor, you should get fairly consistent parameters. It will be slightly conservative in the batch reactor because batch reactors tend to be not as effective in terms of speed as a full-scale reactor because the biomass is not acclimatized, it's not as well mixed, etc. But there's now methods to estimate these input parameters across all three domains. And in fact, we've designed a reactor this size, which was 10 million euros, using a batch test, which was essentially 1,000 euros. And obtained these parameters and then applied them to a continuous reactor. Let's just talk about how you actually fit the data. So here you can see the model, the ADM1, fitted to continuous data over about 3 years. This is from a full-scale reactor in Melbourne, Australia. And the question they had to me was, what's our sludge characterization? Has our sludge characterization changed over time? So I did this process where I fit the degradable fraction, assuming a proportion into carbohydrate, proteins, lipids. So the degradable fraction, as well as the speed based on the volatile solids destruction. And it's got, those parameters are then fixed over this 3 years. So I was able to say to them, this is your apparent degradability, this is your hydrolysis rate for coefficient. So I set the hydrolysis rate for carbohydrates, proteins, and lipids to all be the same value. In this case, about 0.3 per day. And I can say there's no change over time, and here's your values, here's what your performance will be at 16 days versus 18 days versus 20 days, and generate performance curves. And that's a far more powerful way to do design and analysis than the classic method of here's some assumed performance curves. These are actual performance curves based on their sludge. This was a mix of primary and activated sludge. So the other area that's emerged has been co-digestion. And this is just a bit of work from Sergei Stals that he did when he was at UQ. And he looked at basically if I co-digest lipids with sludge and protein with sludge and carbohydrates with sludge, I then get these response curves. You get a very good response of lipids because it's very COD rich. So if I have 1 gram of VS, I get a very good response from lipids, I get a moderate response from proteins, and I get a smaller response from carbohydrates. And then he said, okay, if I put all these together in the ADM1 and I model it, I get this theoretical curve. What happens if I actually mix them together and see what happens? Actually, he gets this curve. And what's happened is that the inhibition effect that's caused by lipids is then mitigated by the fact that you've got carbohydrates and proteins there. But the overall methane yield is the same. So you're mitigating that initial inhibition, but then you're getting the same final yield. Okay, and that could also happen in continuous. So you can look at a control reactor versus one that has carbohydrates added. one that has lipids added, and then one that has proteins added. And you can see with the carbohydrate, you get the bounce that you expect. With the lipid, you get the increase that you expect as you're increasing loading. But with the protein reactor, you only get one increase at the start, another increase when you go to 100%, and then no increase finally. And in fact, it starts to fall over. And that's because you're starting to get this ammonia inhibition. And I just wanted to show this slide because it indicates why we included the additional VFAs. So what you see here is that we get a very fast acetate response when we start getting the ammonia inhibition, and it actually peaks up very high, but then it comes down. But then you see propionate just keeps building and building and building. So the behavior of acetate, which can be predicted by the model quite effectively, is very different from the behavior of propionate, which is not actually included in the model, but we should probably include it, and is different from that of butyrate, which acts like acetate because it's C4 versus C2, compared with valerate, which acts a little bit more like propionate, and it builds up and then doesn't go away. Okay, I just wanted to have a shout out to Xavier's presentation as well, looking at integrated plant-wide modeling. And we got a lot of criticisms when we published the ADM1 that it was too complex, but we found to actually represent all the stuff that happens related to the phosphorus, sulfur, and iron cycles, which are then linked, basically doubles or triples the size of the model. So if you want to really represent what happens in a plant-wide context, you need to make a much bigger model. Luckily, all the processes are still parallel, so we're not changing the conceptual complexity, but we're certainly making the model a lot bigger, just to be able to handle activated sludge systems properly. Why don't I talk about the pH model that much? Well, it turns out that pH prediction in the anaerobic part is not different from pH prediction in the activated sludge part, and neither the ADM1 or the ASM 1, 2, or 3 predict pH very well, or predict chemical processes like precipitation, gas stripping, and other things particularly well. That's why we publish now and have used extensively a generalized physicochemical model. That includes everything that happens in wastewater, a good model for precipitation, a good model for pH, which includes ion activity and ion pairing. That has been a long gestation period, like any task group report, but the model code is available for your use now. Okay, so overall themes are, since we published the ADM1, our increased complexity and demands on modeling. And it's still difficult to generate a unified model. You have to be ready to extend the ADM1 for particular circumstances. There's been a lot more distributed parameter modeling. So modeling lengthwise in plug flow reactors, modeling CFD. And it's a really emerging period of innovation and development. It has been over the last 3 or 4 years, and that's just accelerating. There's a technical report for the CFD working group coming out very soon as well. That should appear early 2022. And that talks a little bit about coupled biochemical CFD models. There's limited scope to generalize excessively. I think the ADM1 is about as general as you want to get. You then specialize the model around that. Of course, there's a place for very simple models, but you need to have some mid-range as well. With that, I might stop, and I think my time's up anyway, and I'll stop screen sharing. Back to you, Angel.
Thank you, Damian. Thank you. This is always a great pleasure to hear you explain us about the ADM1. And let's move to the next speaker, so that as Damian advanced is going to tell us some advances or recent modifications and adaptations of the ADM1. And it's Xavi Flores Alsina. He's joined the Research Center for Process Engineering and Technology at the Technical University of Denmark as Marie Curie Fellow in May of 2013. He finished his PhD at the Laboratory of Chemical and Environmental Engineering at the University of Girona in Spain in 2008, and his research has been mainly focused on the benchmarking, modeling and control of biochemical systems. So, Xavi, please.
Can you hear me?
Yes.
Okay, good. Hello, everybody. My name is Xavier Flores Alsina. I'm a senior researcher at the Process Center, which depends on the Department of Chemical and Biochemical Engineering at the Technical University of Denmark. And the next 15 minutes, I'm going to talk about recent extensions of the ADM1. Just to put things a little bit into context, the ADM1 has been widely used in both industry and academia. As Damien mentioned, had more than 2,000 citations. The model has been extremely successful describing COD, carbon, and nitrogen transformations, but the model does not take into account some of the new challenges that wastewater treatment plants are facing nowadays. And which are these challenges? For example, phosphorus transformations and potential recovery as estruate, for example, sulfur transformation, for example, sulfur reduction and potential sulfite inhibition. And then, of course, like iron transformations, for example, iron reduction from iron 3 to iron 2, and how the iron cycle correlates with phosphorus and sulfur. So in order to include all these elements, so we had to develop a new physicochemical description, also a new biochemical description, and then a new solving routine. So the main idea was to develop a model that was fast enough and strong enough that we could run plant-wide assessment simulations, like, for example, the ones that we performed with the benchmark simulation model number 2. So about biochemical processes. So this is, it has been presented several times today. This is the general schematics of the ADM1. So on top, we have the main or the most, the particulate material, carbs, proteins, lipids, and inerts. And then these are hydrolyzed into monosaccharides, amino acids, and long-chain fatty acids. And then we have production of different types of BFAs and hydrogen through the acetogenic and acetogenesis, reactions, to finally have, methane through acetic lactic or hydrogenotrophic methanogenesis. So the first point that we had to modify here is like what happens with PAOs when they get into the anaerobic digester. So PAOs are still alive, and there is like some empirical evidence that they can still form polyhydroxyl canoates with the subsequent release of polyphosphates. We have also like sulfate reduction equations, reactions, and then where sulfate is reduced to sulfide, and then using different types of electron donors, so hydrogen or BFAs. So if it's hydrogen, we have autotrophic bacteria, and then if it's BFAs, we have heterotrophic bacteria. And then we have also like iron reaction or iron reduction by reactions, and then using different types of electron donors. About the physicochemical description, so this is the original speciation model. So in order to improve that, so we had to go to full description of ions. So we had to increase the list substantially. And then, of course, we had to account for multiple species. And then, how these species, they also can link together, like forming like different types of ion pairs, for example, with aluminum, calcium, iron, magnesium, etc. So the main idea is to predict pH, but taking all these elements into account, ion speciation and pairing, and then running these calculations under non-ideal conditions, so continuously tracking ionic strengths and activity corrections. So on top of that, so we had to describe precipitates as well. And then we identified the list of compounds, like using the list of ions that we accounted for example, carbon and calcium. We have like formation of calcite. Then ammonium, magnesium, and phosphate, we have estruate. And then up to a list of like 13, 14 compounds. So new biochemical description, new physicochemical description. And then we invested quite a lot of time on solvers as well, because the resulting model that we had was very stiff. So we had, very slow and very fast processes. So for this type of systems, normally use stiff solvers. But as I mentioned at the beginning, so we wanted a model that was also like useful for control. And then some of the blocks that we want to use for control, for example, like buffers, delay blocks, noise. I mean, all the stiff solvers that they don't work that well. So we had to go for non-traditional, or more traditional, like stiff solver, non-stiff solvers, because these ones are like more appropriate for control. But of course, so they cannot handle stiff systems. So we had to develop a new numerical method based on a multidimensional version of the Newton-Raphson. So we had, a new biochemical, a new physical chemical, a new solving routine, and we produced quite a lot of scientific outputs. So there was a very exciting times for us because we got so many papers, and also because there was so much to be done. But at the same time, we got a lot of criticism because most of the work that we did was very theoretical or only applied into virtual appliances like the BSM2. So that's why in 2016, so we embarked in new projects, trying to validate these models on real systems or full-scale plants. And then the first person that was brave enough or stupid enough to do that was Hannah Feldman, who was a PhD student in our group from 2015 to 2017, in collaboration with Lund University, also University of Queensland, and the university and the company Novozymes. And what we tried to do is to use this modified version of the ADM1 to describe, an anaerobic granular sludge reactor. So this is a general schematics of the plant that we tried to model. So the wastewater comes from the bottom, and then there is a mixing section, and then goes to an expanded sludge bed where the wastewater is mixed to a granular sludge. And then we have two separation modules, upper and lower. And then on top of that, we have a gas liquid separation where the biogas is sent to a gas turbine for production of electricity and heat. And the liquid is recirculated to the bottom again, to the mixing section. So in order to model this type of reactors, so we used what we call a multi-scale approach, where we try to describe the hydraulics, combining different types of continuous stirred tank reactors. And then we assume different types of granular sizes. So we assume the bigger granules in the bottom, and then the smaller granules in the top, just to mimic the effect of gravity. And then we went for full biofilm modeling. So we described, the movement of solubles with diffusion and the movement of particulates through convection. So this is a general schematics, how the biofilm growth was described mathematically. So we had on one hand, precipitation, formation of different types of inorganics. And also we had the growth of the different types of biomass. And then in the middle, we had the weak acid-base chemistry that was the glue that maintained all these elements together. So this is the model. So we tried to validate this using two different data sets that we collected at different measuring campaigns. And then we could see here, in black, we have the plant measurements, and in gray, the simulations. And then for different compounds, methane, CO2, H2S, ammonia, phosphate, sulfate, BFA, COD soluble, COD particulate, pH, calcium and magnesium. So basically we were able to describe the main processes taking place in the anaerobic digester: hydrolysis, acidogenesis, acetogenesis, methanogenesis, sulfitogenesis, nitrogen and phosphorus release, weak acid-base chemistry, and ion behavior. So we use most of the 4 parameters for that purpose, and we had a 10% deviation between measurements and simulations. We had a second data set as well. Black, we have simulations, and black, we had the measurements, and gray, we had the simulations. And again, for methane, CO2, H2S, ammonia, phosphate, sulfate, BFA, COD soluble particulate, pH, calcium and magnesium. And then again, so we could describe most of the biochemical processes considered in ADM1: hydrolysis, acidogenesis, acetogenesis, methanogenesis, sulfitogenesis, nitrogen and phosphorus release, weak acid-base chemistry, and ion behavior. And the deviation was also very low, 11% between measurements and simulations. So we have a calibrated model now. So what do we do with it? So we try to run what we call optimization studies. and try to see, for example, if we can make predictions and use, to make decisions, for plan Y, for the system we are trying to describe. So one of the things that we found out is that using the model that we could reduce by 40%, like the use of chemicals, moving the pH from 7.2, that was like the current operational mode, to 6.8. And also at the same time, we were ensuring, a lower precipitation level in the reactor. That we could reduce, like the use of chemicals, like promoting CO2 stripping in the pre-acidification tank before the anaerobic digester. And then also like something quite interesting is that we could assess, for example, what would cause, removing the sulfate before the reactor and the benefit that we could have afterwards. And then, what the model told us was that basically it didn't compensate, like the cost of treating the sulfur at the beginning for the benefit that we got afterwards. So we did also like some other things, so we tried to model like, how the different precipitates that were accumulating in the granules. And then what we found out is that we have considered different granular sizes, like the bigger ones in the bottom and the smaller ones on top. And then that this, like the precipitates were accumulating in the outer parts, for example, in the bigger granules, and then in the inner parts in the smaller granules, which basically was due to mass transfer limitations. And this is, what the model was providing us, and then we validate that in, using like taking samples from the reactor at full scale, and of course we could see exactly the same behavior. And then we use the model as well to evaluate the long-term effects of intergranular precipitation. So we assess, for example, what would be the impact of using, because it was like an industrial plant, so the effect of using a very alkaline stream in the anaerobic digester. And then what we found out is that we had formation of precipitates in the granules, and they could be competing for the space. And at the end, we had, like a catastrophic event on that digester, basically accumulation of BFAs and a reduction of the methane. So just to finish a bit, we believe that sharing is caring. Thanks to the help of these 4 lovely men, so we developed this platform, GitHub, where we have uploaded, all the models that we have constructed during the last 15 years. So please check, and then you can download and see, for example, most of the studies that we have conducted. And then just to finalize, my presentation, some important things that, that we found out was that the case studies that we have conducted, this was the first one, but we have like 3 more published and then 2 more ongoing, has contributed enormously to gain credibility in our models. We realized as well that the process of constructing the model and taking the samples from the plants and calibrating was the main learning experience. And most of the simulations that we have conducted confirms just what process engineers already know. But of course, in some other occasions, they can provide with some surprises, for example. For example, when it comes to assess the capacity of the plant. And then we are very happy because the GitHub page that we have launched at the beginning of the year has been extremely successful. So we had more than 1,000 downloads. So please, like check and see if it's something useful for you. So that was all from my side.
Thanks, Xavi. Well, I have to get you the control, or if you can stop sharing, could be easier. Thank you. Thank you. There is a lot of work, of course, on trying to make the most for catching the reality of the systems through the extensions of the ADN1. So it's a huge work that has been done, and thank you, Xavi, for all of this and all the co-workers. And let's move to the last speaker, that is Jan-Philippe Steyer. Please, in this point, I want to remind you that don't use the the normal chat function, please use the Q&A to put and write your questions, and we will answer it at the end of the session. And well, it's a pleasure for me to introduce Jean-Philippe Esteller. And he is, well, as most of you know, is research director at the INRAE since 2004. He's a very renowned expert on the field of AD instrumentation, modeling and control, and he's also an expert on other bioprocesses, such as biohydrogen production, wastewater treatment, including microalgae cultivation, and generally on bioprocesses applied to resource recovery. So Jean-Philippe, please, the floor is yours.
Thank you very much, Angel. So good morning, everyone. And thank you for all the organizing team to have organized this webinar. So the topic of my presentation today is about advanced instrumentation for automatic ADM1 parameter calibration. So I will say a few words about how could we predict biodegradability first. So the first thing is
Jean-Philippe, can I interrupt? Can you go into full presentation mode on your slides?
I am.
Not here, no, only locally apparently.
Is it good now?
Maybe if you have two screens, you are sharing the second one and not the first one, and you're projecting in the other one.
No, I have only one screen. Well, it was working before. I'm sorry. I don't know what to do.
No, okay. If you cannot solve it, Jean-Philippe, just go ahead with.
I'm very sorry for this. It was working well. So, which slide do you see? Do you see the second slide?
Yep.
Yeah. So you have different ways to predict the biochemical methane potential. It's either you run a BMP test, which will last for 30 days, 40 days, and you have the results here, or you can make an infrared spectra. Here you have an example of infrared spectra in the near infrared range from 1,900 nanometers to 2,500. And the idea is to link with a model these two things. So the way you do it, it's with PNS model, where you want to predict the BMP value based on the spectra of the waste. And in order to do this, you have to make a model, and you have the B coefficient here, which will multiply each absorbance of the wavelengths of each wavelength by a number. And here you have the B coefficient of the model, and you can see that it's varying with the wavelengths. And what is nice, it's when you have, like here, around 1,917 nanometers, it's the wavelength of the lignin, and you see here that the B coefficient is negative. Whereas for the others, like starch, it's positive, so which is an indication of the biodegradability of each part of the waste. So what we did is like more than 10 years ago now, we developed an approach to calibrate, to predict the BMP values based on the near infrared spectra. And here you have the results over more than 500 substrates that can be predicted quite nicely with an accuracy of 10 to 20%. So we wanted to go further. So what we did is we take quite big numbers of samples: grease, oil, fruit, vegetables, meat, fish, microalgae, sludge, and slurries. And here you have the values of proteins, lipid, carbohydrate, COD, and BMP values of each of these 340 samples. Sorry. And so what we expected to do first is to build a model that, based on the near-infrared spectra, could predict all of these values. Unfortunately, we were not successful. And so what we did is we built one model for each. So one model for the BMP prediction, one model for the carbohydrate concentration prediction, etc. And this works quite well. Here you have the results. The protein here, lipids on the right, carbohydrate, and then COD. In the green crosses are the calibration of the model, and the blue diamond are the prediction, the validation of the model. So you can have quite nice results. And we wanted to go even further. So we wanted to be able, and I have to admit that I was not, I didn't think it was feasible to do this, but we wanted to predict the kinetics. So the first thing we did is we modified the BMP test. And instead of running a BMP in a bottle, like a classic, it is classically done, we run in a reactor of 5 liters, and we with a low value of VS at the beginning to avoid inhibition. And we ran a batch test for a few days. And after we iterate this, so we run a second batch and a third batch, etc., etc. It could last for 40 to 60 days, so it's quite long. But at the end, what you get is you converge to the same BMP curve, which shows that the adaptation of the inoculum to the waste was successful. So we could use this last value of the batch test as a good sign for the kinetics, and we could model it. So you have the BMP here value, which of course is always the same, and you have the kinetics that can be used for modeling. So in order to predict this curve, we did several attempts to do it, and what we did at the end is we took the percentage of the BMP on the y-axis at the time, and we divided by different percentage of the BMP reached. So at the end, you end up with 19 points. So you have the time for 5% of the BMP, time for 10%, etc., up to 95%. And so we built 19 models to predict each time of the BMP achievement. And this is the results.
Jean-Philippe, I don't think your slides are are following your presentation. You're still showing your slide number 6 on.
Wow, I'm sorry for this. I'm really sorry. So you didn't see all these slides?
No.
Oh. I don't know what.
If you go out of presentation mode on your local screen, then maybe at least you see the same on your screen as we see online.
Yeah. So so do you see the slide 7 now?
Yep.
So it was what I was saying. It was we wanted to predict all of this BMP, carbohydrate, proteins, and lipids. With one model, it's not working, but with 5 models, one for each component, it was working. And these are the results for proteins, lipids, carbohydrates, and COD. The cross is as a calibration of the model, and the blue diamond as a prediction of the model, the validation of the model. What we did after, it's the kinetics. So I said that we modify the protocol of the BMP. So we run it in a reactors of 5 liters, and we do several batches, which last each about 1 week to avoid inhibition. We have a low value of VS per liter, and you iterate this batch test until you reach the same BMP curve, which means that the adaptation of the inoculum is okay. So you can use this value to model the BMP. And that's what we did. So we separated the value of the maximum BMP you reach, and by 5%, 10%, 15%, etc. And we developed 19 models for all the time, to predict all the time of reaching the BMP value at the end. And it worked. So you have here the predicted time over cross-validation and validation. You could see that you have quite nice results. And we use this value to predict, to calibrate the ADM1 parameter to feed the prediction of the near infrared value predicted by near infrared. And you have here in gray the curve predicted by the near. In purple, you have the ADM1 with standard parameter without any calibration. And after calibration, so this is automatically done with a procedure we developed. What you can see is you have a fitting of your curve, which indicate that the ADM1 was able to model this. And this is fact, it's at the end, what we get, it's from the waste. First step, you have to freeze drying and grinding the waste to have it homogeneous and to remove the water. And you have the spectra after. And from these spectra, you can predict all of this, all of these parameters in less than 4 days. 4 days, it's because this step, the freeze drying and grinding, takes most of the time. This step is very fast. And if you're interested, this work was part of the PhD of Cyril Charnier, and you have 2 papers describing all this methodology. And I will just finish saying that while it works, there's a company that has been created called BioIntech, and they have now like few years experience of applying it at a full scale. And you can see here that if you have a good characterization of the waste, you can have a very nice following of the dynamics of the process. And this is full scale, and they applied it for about 50 plants now. Well, again, I'm sorry for the problem with my sharing. I don't know what happened. It was fine just a few minutes before the webinar. Well, thank you very much.
Thank you, Jan-Phil. It's just life issues, so don't worry. Well, let's move to the Q&A section. And we have some interesting questions for all the panelists. And well, I will start more or less in the same order, and the panelists have to speak. So the first question from Sign Up is for Raul, and well, thanks for your presentation. And there is 2 questions regarding the phenol degradation in anaerobic membrane bioreactors. Let's start with the first one. That is, was there any measurement of toxic metabolites of phenol in the reactor?
Yeah, I kind of reply, Sign Up before, because I mean I provided an overview of all the presentation, but I don't have specific details for that. But I guess, I mean, Jules Van Lee is a very good group. I'm sure they check for metabolites to make sure that they have full mineralization of the pollutant. And so, and also she also asked about the reduction in toxicity levels in the anaerobic membrane bioreactor. And I guess that when you have a complete or much better mineralization of the pollutants, the toxicity levels will decrease in the cultivation broth. So I think it's that's probably the answer that Jules would provide in this case.
Yeah, there was also the second one, but well, it's in the same line because actually there was a summary that the question was, did the phenol level increase when membrane bioreactor was used? So I guess that it is about the concentration. I don't know if you have any remark on this or not, or
yeah, it's probably, I mean, if degradation is much more efficient in anaerobic membrane bioreactor, toxicity levels would decrease consequently.
Yeah, I have a personal question about the summary. Just it's a general one because I'm very interested in this issue, and I don't remember if there was some remarks about the cost of recovering all the resources. For example, when there was the speaking about the nitrogen and phosphorus recovery, etc. There was some remarks about the life cycle assessment and life cycle costing or something similar.
Yeah, I think the main remark from Anna was the amount. The problem is that the market driver, the amount of nutrients recovered, and the are not so big for fertilizer companies. So they are not very much interested so far. Because the quality is not it's not like high quality, and also the production is small for the amount that the fertilizer company use. So things right now is more like a market driver, and apart from a technology limitation in nutrient recovery.
Yeah, we should wait for the complete depletion, so in the resources for increasing this value.
I mean, it's true that the price of chemical fertilizer has doubled or even tripled over the past month due to increase in electricity. So, I mean, scenarios can rapidly change, and also she point out the need to look for local solutions rather than, maybe shipping the recovered fertilizer to 500, 600 kilometers so probably there should be a balance and at local level.
Yeah, that's true. Zero carbon value chains.
Yeah.
Well, Damien, I have one question here for you, Damien. Well, I read it directly. I just wondering if in your research you work about gas modeling, especially H2S, and if we can control production of H2S by dosing some salt of ferrous of iron. Also, we can control soil precipitation bias at the same time, and as well as dosing. Will impact the normal performance of the digester or not?
Yeah, so the chemistry model that Javi has put together, along with many other people, of course, as he noted, is highly capable of this. So it will predict struvite, and in fact, there was another question about fitting rate parameters. And in general, you have three scales of process. So we did a lot of work applying the chemistry model in different applications across plant-wide. You have these three speeds of process. You have FES, which is very fast, on the order of minutes. You have struvite, which is on the order of hours, and you have calcium carbonate, which is on the order of, and most of the minerals fit into these three categories. And as long as you get the right order of magnitude, then you get reasonable prediction. And you'll see, for example, calcium phosphate forms very slowly because, and you will form first FeP, which forms quite quickly, but then that will transit to calcium phosphate as you get this dimorphous crystal forming. So it's the rate parameters that we've provided in the papers, particularly those by Christian Kazadi, can be used directly. And the models we use a consistent precipitation model now, which considers the order of precipitation correctly. So this is actually almost no parameters to be fitted in precipitation or chemistry at all. You just apply the model. It's a very complicated model, but it doesn't need you to fit parameters.
I don't know, Xavi, if you want to add something of.
No, I just, yeah. So, I mean, the number of contestable parameters are very low because, I mean, it's based on thermodynamics. So, it has a high degree of complexity, but I mean, the number of calibration parameters is rather low, actually.
Mm-hmm. Well, and in line with this, well, I have another question from Gabriel. And of course, thanks for the great presentation. And well, some general questions. After all these ATM1 modifications, where do you see the field of AG modeling going? What do you feel are the most important points yet to be targeted? This will be the first question, maybe for Damien and for Xavi.
Damien?
Yeah,
Where we are going?
well, I think
Just passing the
one
hot
of the things
potato.
we were asked a lot was, should we present an ADM2? And really what you have is a lot of ADM1. So you have the ADM1 of Xavi, which has got what at the moment, something like 60 states. But you have the ADM1 of Jules Van Leer, which is working well with anaerobic MBRs. You have the ADM1 of the granular team in Delft, which is focused on granulation. So there's a lot of forks, and they're all equally valid because they're all applied to different applications. And you have the ADM1 of Jean-Phi, which has got 10 ADM1s together that all represent different speeds of processes and different microbial communities. So it's supposed to be a research tool, a common basis, and that's what it is. It's not supposed to be the end result. And in fact, the ASM1 has been modified extensively, for example, for use in algae conditions. So the bioalg model combines an algae model with the ASM1 and modifies the ASM1 quite heavily as well. So that's the way these models should be used. They shouldn't be used as the end result, but there's no application, there's no one generalized application. You can't just say this is the perfect model for every application. It has to be, otherwise it would be too crazy big, and it would include a lot of stuff. And in fact, all the commercial packages that focus on sludge modeling have used reduced forms. None of them have become particularly more complicated.
Yeah, you have already answered one of my questions, that was ADM1, yes, ADM2, yes or not, but it's clear. So, and a second question also is knowing that the web model worked, what do you think is missing to increase their usability by industry players, if there is something or not to be taken into account for increasing this applicability? I don't know if Gabriel is focusing on the selection of key parameters to be calibrated, or some guidelines, or some go-to modeling practices, or I don't know. Damien or Xavi?
I think the input characterization issue is still not well understood. I've tried to present it as well as I can, but it's always a problem for people to use input characterization, and that's the primary question I get answered. You know, we have 19 states. Do we have to have a value for all of them? No. You need to have an understanding of the system to characterize the input. So that's the biggest hurdle that people have in general. Then there's just simply the fact of being able to model, and that's a set of skills that you develop as a chemical engineer. And all the people on the panel have developed those skills, but it's taken us a while to develop them. And part of that is training. So the MIA and other IWA groups organise training as part of these conferences. And when we have face-to-face again, we'll start. I've presented many training sessions, and I'm happy to present more. And I'm sure the other panelists are as well, have done so, and will do so again.
Xavi, I have a question here. Were you able to properly model the inhibition of methanogenesis caused by this precipitation process over the granules?
Yeah, I tried to reply on the chat. You know,
Yeah.
we did not actually.
I mean,
because we didn't have like enough empirical evidence. I mean, the only thing that we could account for was the competition in the granule between the biomass and the precipitates, which basically washed out the biomass, and then of course you have a reduction of the methanogenic activity. You know, but you know, like inhibition of calcium carbonate as such, I don't know if this thing does exist, you know, but I mean, we did not.
Okay, I will move now to Jean-Phi. Well, I have a first question from my side. Maybe this is a tricky one. And I was thinking, well, there is a lot of work for calibrating the near spectrophotometry. And I always thought, or I always was wondering if, for example, if the reproducibility of the results will be also as well as the ones that you have when you go to other substrates. But from different regions and different conditions, not in France, for example, getting some substrates in the area. The results will it will be feasible to reproduce the results.
From our experience, the reproducibility of the infrared spectrometer is better than the BMP itself. And it will be difficult to predict something you don't know.
Mm-hmm.
So you can make like a nearest neighbors approach, or like classification of the spectra for a substrate you don't know, to predict it. But the best way would be to have it in a database. So we got quite nice results because our database is quite huge. Like now we have 1,000 substrates that has been fully characterized, and this makes the robustness of the approach good.
Mm-hmm. Well, thanks. And well, there is also Roman. Roman, you know Roman, and he's he has a question. He say that even if the repeated BMP converts to a unique CH4 methane production curve, can we really extrapolate the kinetic values to actual digesters?
I would say yes, until you always in a easy mode. I mean, if there is an inhibition, for example, it would be difficult to extrapolate. So when everything is working fine, I think you can extrapolate. If you have inhibition and you manage your process not the correct way, maybe you will have it will not be possible to extrapolate this.
I'm focusing on the size, because he says that the BMP are usually carried out using the low substrates concentration, and the microbes might be not the most representative ones from the microbial community observed in the full-scale plants. If you have some feedback on this topic or some experience on this, just for the microbial community, not for the inhibitors.
That's something we are working on now, because it's difficult to find data with operating conditions, measurements, and at the same time, nice microbial characterization. So that's something we're working now to make the link between all of this. But so far, no, we don't have.
Good. And also related to this, Christopher says to Jean-Philippe and to Damien, I wonder if it could be more efficient to generate a standardization to predict the maximum methane production with BOD type test, but not with the biomethane production tests, because this test could be shortened and faster than the BMP test. Do you consider that the dynamics shape change of the BMP curve can give us hidden information to model the reactor at full scale?
Damien, if you want to answer, I mean,
I don't believe in short BMPs. It's not a. There's people who disagree with me, and they've got valid points of views, but I don't believe in short BMPs, because from what I've seen, if you operate a BMP properly, it will take 10 to 20 to 30 days. I haven't seen that the early stage performance is a good predictor of the final. It follows an exponential decay curve, but not exactly. You can't predict the final BMP from the performance in the first 3 to 5 days, particularly for activated sludge, where you've often got a rapidly degradable and slowly degradable fraction. And Jean-Philippe has done probably more BMPs than we have, and we've done a lot. We've done over 5,000. And we see, particularly for activated sludge, you can get a very long tail, and we have to then split it into this rapidly and slowly degradable fraction. But that's my opinion. But there's certainly people who advocate for short BMPs.
Jean-Philippe, do you have something to add, or Jean-Philippe?
I fully agree with Damien. You cannot predict the BMP from the beginning of the curve. In our case, what we did is we reduce we put condition that BMP is reduced, but at the end you make several one, so it lasts the same amount of time, and you always reach the same value as a classical BMP. So, but it took us quite some time to set up all these procedures, and it's published if you're interested.
Yeah, if you do pulse BMPs are different, where you're continuous pulse adding. That's a different, but that is also a highly skilled procedure, because when you mess with a BMP, you're changing the conditions as well. So you have to know what you're doing. Yeah.
Good. Ulf, I don't know if we have time for a couple of questions more, or
I think we have now concluded the 90 minutes and we are on overtime. So maybe it is time to, I see some participants are also starting to drop off. So I think it might be a good possibility or a good opportunity to go out of this webinar. Sorry. Just before we end, I want to say great thanks to all the presenters and also to the audience of the webinar. Thank you for your questions. I would also like to give a very special thanks to Gabriel Capson-Torjo at the University of Santiago de Compostela in Spain, who has been very much involved in the whole process of organizing and setting up this AD webinar. So thank you, Gabriel, if you are listening. We also hope that you've enjoyed the show, and hope that you will also join for our future webinars. At this point, we have the holidays coming up and so on, so we don't have a specific webinar pinpointed yet, but probably in February 2022, the next webinar will be online from the Modeling and Integrated Assessment Specialist Group with a topic to be decided. And also remember that if you have a need for setting up or creating a webinar on a topic of your own, which is of interest to modeler with regard to modeling and control issues, then you can contact either some members of the management committee of the MIA group, or you can contact the management committee of the ICA group. And we will try to assist you and make your webinar come true in the close future. And please also remember, if you're not an IWA member, then please consider to become one. And then you can also enjoy all the benefits that such a membership will provide for you. And with that, it is time to say thank you, and a great start for this week, and hope to see you very soon again. Bye-bye.
Thank you. Bye, everybody.
thank you all.